Introduce you, Professor Nick Martin.
Well, thank you very much, and thanks for all turning out at this hour when you probably would rather be at the bar or having something to eat or something like that. But I appreciate your dedication in being here and wanting to broaden your minds a little bit by hearing this. Now, because it's this hour, I've actually tried to make it fairly light-hearted, so please don't think this is as serious as my science gets. I've tried to give you the light version for the evening. But somebody asked just when I came in, where is QRMA, by the way? And I suppose I should have put one of those boring slides of my building or something. But anyway, it's at the Royal Brisbane Hospital, just north of the city, 8 kilometres away from here. So what do? I study twins. Why do I study twins? Not because I particularly like or am thinking about twins. It's because they're a most fantastic model for it's the most wonderful natural experiment we've got for understanding the causes of individual differences between people, individual differences in every aspect of being human. So most people just focus on twins as nice photogenic things for magazine covers and so on. But you know, identical twins are incredibly similar. Even their teeth can be exactly the same. If you're a dentist and you look at these imprints, you're absolutely astonished by the detail at which these twins are the same. Every little cusp is the same. And they even do bizarre things like marrying other identical twins because, you know, they're attracted to the same genotypes. But not all twins are identical. Those are the ones that get most attention. Of equal importance to us as researchers are non-identical twins who share only half their genes in common. And in fact, the whole twin study depends upon the comparison of identical twins who are identical because a single egg is fertilized by a single sperm, and at some time very soon after fertilization, for reasons that are not understood, the zygote undergoes spontaneous fission. Usually, well, we don't even know exactly what cell stage, but putatively at the 8-cell stage or 16-cell stage, it undergoes a fission. So you have these two identical zygotes then growing up in the same uterus and born and raised at the same time. Now, of course, all sorts of things can be freaky about the twin situation, so it's very fortunate as scientists that for that we have a control situation of non-identical twins or fraternal twins who in fact are just like ordinary brothers and sisters, except for the fact that they are caused when 2 eggs are released in a single cycle and fertilised in a single cycle. But they are then gestated and born at the same time, so they form the perfect control for identical twins, much better than ordinary brothers and sisters. They are just like ordinary brothers and sisters, except for that freak of their conception and gestation and birth. But and they come in the same ratio of a quarter boy-boy, quarter girl-girl, and a half boy-girl. So the fact that these two have such similar origins and then upbringings, because most of them, the vast majority, are reared in the same house, means that these form a very nice control for each other in the study we want to do. And just in case any of you get confused about people, I sometimes have twins. I've had twins walking into me saying, we're identical twins because there's only one placenta. One of them has got blonde hair and one of them has got black hair. Or conversely, you'll find non-identical twins who swear that they are identical because they shared the same placenta. Or so rather, you'll find identical twins who their mothers can't tell apart, but will tell you that they're non-identical because there were 2 placentas. In fact, there's a very poor relationship between placentation and whether the twins are identical or not. I won't go through the detail there, but you can see the numbers that you do not use the placentation of the twins as a guide to whether they're identical or not. The most reliable way to do that is to do genetic testing, and this is using microsatellite markers for our friend in the front row here who works on them. No, she works on something else. But yes, no, she does work on microsatellites. So this is the way that we test for zygosity, and here you can see a pair of identical twins, and we're typing nine genes with what are called microsatellite markers that are colour-coded for your convenience, and you can see that they're absolutely identical at every one of these. And here we have two pairs of non-identical twins, and you can see that some genes they have are in common, but others, like this pair here, are different. And in fact, if you actually count them up, about half the genes are the same, half the genes are different, and that's exactly what we expect from the laws of Mendel. But this is the most reliable way of telling whether twins are identical or not. Now the twin design, the comparison of identical and non-identical twins, is so popular that it spawned the existence of twin registers all over the world. I edit this journal, and we just published a special issue on twin registries of the world, and I think we had registries from every continent and I think 78 countries where people have founded twin registries purely to help with research. And so here are some data which actually make the point as to why they are so interesting. Compare the correlation of, so what you've got here on all of these slides, these graphs, is the value for twin 1 on the x-axis, the value for twin 2 on the y-axis, and here we're looking at height, and we see this extremely strong correlation in identical twin males, and in non-identical twin males you see a lot more scatter, and you see the same pattern in females, and the DZ correlation much lower than the identical twin correlation. And here we see the same sort of thing for brain volumes of twins. We see this extraordinarily high correlation for the identicals and much looser correlation for non-identicals. In fact, we can quantify this a bit more. This is looking at blood cholesterol, and we're looking at a correlation of 0.86 for identicals, 0.46 for non-identicals. And in fact, a little bit of simple algebra on the back of an envelope, we just subtract, because identical twins share all their genes in common, non-identical twins share only half their genes in common. You can just subtract the DZ correlation from MZ correlation and double it, and that gives you what's called the heritability, or the proportion of variation in the population, the proportion of individual differences which are due to genetic effects, and in this case about 80% for heritability. So this is a useful thing to know. I mean, everyone I think is interested in individual differences in how you look, in your health, in your personality, your behaviour, also your disease proneness for common diseases like asthma or eczema or depression or something like that. And using the same principle, we can get a very rapid fix on the relative importance of genes and environment. So most of the work that I do anyway, it depends upon using identical twins who are born into regular households and reared in the same household, just like most of us have been. But occasionally we get these sort of freakish circumstances that cause twins to be separated soon after birth and reared in quite different environments, often usually completely unknown to each other, and then finding each other later in life. Now this provides an even better natural experiment for the researcher, and this is just one example of a pair of twins who found each other and were reunited. In fact, I found a series of these in Brisbane. I was giving a talk just like this to, I think it was a Rotary Club or something, and talking about how valuable these sorts of twins were. And afterwards, this woman came up to me and said, "Oh, I'm just like that. So I just met my identical twin sister." I said, "Oh, fantastic. You know, how did that happen?" She said, "Well, I work in David Jones, and I'm the fashion buyer for David Jones. And at a certain point, all the reps started saying to me, "What are you doing, Michelle? Are you too tiny or something? I just saw you at Myers." And she said, "What? No, I don't work at Myers." After this happened the third or fourth time, she decided to investigate a bit more closely and went over to Myers and found a woman who looked exactly like her, doing exactly the same job as her, and turned out to be her identical twin, from whom she'd been separated 37 years before. And they'd grown up, one in England and one in Brisbane, and the other one had ended up coming to Brisbane through a circuitous route. And there they are. But how amazing that, you know, after all that, they should end up doing exactly the same job. And this is the sort of story of these identical twins. There's the fellow called Tom Bouchard in Minneapolis has made his life's work studying these twins and flying them in from all over the world, and he ended up collecting about 80 pairs from around the English-speaking world, and flew them in, and about half of these pairs actually met at Minneapolis Airport, flying in for the first time. And the stories about them arriving and wearing exactly the same clothes and the same number of rings on their fingers and having the same hobbies and jobs and so on. One of the first things he did with them was just ask them to stand against a wall and take their photograph. And this is sort of quite neat because the top three pairs are identical twins, and you can just see the way they naturally adopt the same stance, you know, in standing. They do the same things with their hands and so on. Whereas at the bottom you see the non-identical twins who have quite different styles. And it's a trivial thing, but it just sort of makes the point. And there are lots more examples like that. And in fact, in case you think this only pertains early in life, these are Swedish identical twin pairs separated really early in life and united very, very much later, and even into their 80s. Are still showing the most amazing similarity in their aging patterns, you know, but both things going bung at exactly the same time and those sorts of things. So genes seem to affect many characters right through life. And even looking at behaviour, these are correlations for 3 different aspects of behaviour in identical twins read together, and in this smaller set of identical twins read apart. And although the correlations are not exactly the same, and the first one for positive emotionality is a little bit lower for the twins read apart, it's not significantly lower, and you can see that those correlations are pretty damn close. So unfortunately for the sort of Freudian psychologists, it doesn't really suggest a huge influence of family circumstance or family environment, because these people have been brought up in quite different circumstances, yet apparently are about the same as if they'd been brought up in the same house. What is the value of twin studies? I mean, it's fascinating stuff and everyone likes to hear it, but I mean, it actually does have a serious influence on research agendas and how people look at disease and how people look at the world. And I'll give you 3 examples. When I was your age, that's an appalling sort of thing to say, I know, but when I was your age doing psychology, I mean, the prevailing theory, that was in the 70s, the prevailing theory of autism was that it was caused by cold mothers, by having a cold mother. And there are all these environments, sort of, you know, frankly crappy environmental theories out there about how, you know, terrible cold mothers caused autism. And then somebody came along and did this twin study, which actually by the standards of today is very small. They just got 11 pairs of identical twins, where one was autistic, and looked at the other pair, and of those 10, both had autism. Whereas in the same number of dizygotic pairs, only 2 were concordant for having autism. And that's telling you, in fact, you can do the maths and show that that's consistent with a huge genetic influence on autism, at least 80%. And that has completely changed the research agenda for autism. It's also been incredibly liberating for mothers, you know, who, you know, previously felt incredibly guilty. Oh, it must have been because I was so cold. It actually turns out mothers of autistics aren't cold. And you know why? Because they've actually got half those genes already. They're already heading down that path. And probably what's causing autism is the combination of this sort of tendency that way from both sides of the family. I mean, the place where autism is most frequent is Silicon Valley. You get all these semi-autistic nerds marrying each other and having kids. So another sort of case history is